Hey everybody, this is going to be the video for chapter 43, which is all about the immune system. So to get started, the immune system is the system in your body that protects you from pathogens. So pathogens, these could be viruses, these could be bacteria, it could be just like dirt or debris, or it could be parasites. So all the stuff that makes you sick. So the immune system is our way to protect from these pathogens that we might take in from the environment. And so there are sort of three ways to sort of help prevent this. The first way would be to create a barrier. And a barrier would be something that's stopping pathogens from getting into your body. So this would be like your skin or your mucous membranes. And then the next two processes are recognition, which means recognizing that there is one of these things present, and then a response to hopefully destroy these things. So you have two types of immunity. We have one that's called innate immunity, and we have one that's going to be called adaptive immunity, and we'll get into that one next. But for now, we're going to talk about innate immunity. So innate immunity you have from birth. When you are born, you have your innate immunity. It is going to be occurring in every animal, so all animals have this. It has multiple components, and it's sort of the rapid response because it's always kind of there. It's very generic, so it's also non-specific, you could say. So then when we're talking about innate immunity, we have two sort of subcategories. We have the external innate and the internal innate. The external innate is super, super easy. First is you have skin, and skin has this oily surface, and obviously it is a physical barrier. And this oily surface plus the actual barrier both prevent entry of pathogens. Then we have mucous membrane, so you have the mucus in your throat or in your nose, and that's actually to protect you from pathogens. The mucus is actually able to trap pathogens, so it's going to sort of block them because it's very viscous, right? And then you have these cilia can sweep away pathogens, and cilia are these tiny little hairs in your mucous membranes. So both of these are just preventing pathogens from entering the body. But the sort of big weakness of the external innate immune system is you can get wounded. You could get a little cut on your finger that bacteria could then enter. You could ingest something, so you could swallow something that has pathogens in it, and then it can make its way in your stomach and intestines. So that's sort of the first line of defense. Then you have your internal innate immunity, which is going to be all about your leukocytes, which are your white blood cells. And remember, these are going to be made in red bone marrow. So the types of leukocytes that we're going to talk about, there are four main ones. We have macrophages, neutrophils, eosinophils, and dendritic cells. So macrophages are very important for signaling. They release these things called cytokines, which sort of signal all of the other immune cells to sort of come and help out. Macrophages can also present antigens, and we'll get into what that means later when we talk about adaptive immunity. But then the big thing is macrophages can phagocytote, which means to engulf pathogens. So if we have a dangerous little pathogen here, and we have this macrophage here, this macrophage can move over here and actually sort of start to engulf it until eventually it fully digests this pathogen. And then once this pathogen is in the macrophage, it can be degraded by enzymes in the macrophage. Neutrophils do the same thing. They phagocytote bacteria, but they're really, really fast at doing it. They're faster than the macrophages are. So neutrophils' only purpose is to phagocytote bacteria. Eosinophils, if you see eosinophils, I just want you to think parasites. So eosinophils are the leukocytes that are going to target parasites and kill them and get them out of the body. And then the last one, we have dendritic cells, which are antigen-presenting cells, or APCs, which we'll talk about again later. And this is sort of how we bridge the innate immunity to the adaptive immunity. So let's talk about this word antigen. So let's say we have a pathogen. Let's say that this is a bacteria. On the surface of this bacteria, there are going to be a bunch of little proteins that are called antigens. And the antigens are what allow the immune cells to detect and recognize this pathogen. So that's what antigen means. All right, so the next thing I want to talk about with our innate immunity is the inflammatory response. So the inflammatory response is going to start when a bacteria or pathogens enter a wound. So I have a little nick on my hand. Let's say we have a little nick on the hand, and bacteria are going to go into that wound. And once bacteria enter that wound, it's going to start the inflammatory response. So one thing that's going to happen, and this is going to happen anytime you have any kind of wound to your skin or any kind of external bleeding happening, is your platelets in your blood, which hopefully you remember, we talked about those last chapter. The platelets in the blood are going to release blood clotting proteins. And what that's going to do is just slow and stop the bleeding. And it's also going to make the blood harder to travel through for those pathogens entering the wound. So the next step in the inflammatory response, the bacteria entered, the platelets have clotted the blood. Now our tissues and the macrophages in the tissues, remember macrophages was one of those leukocytes, those are going to release chemokines. And chemokines are a type of cytokine, which essentially that type of molecule signals other immune cells. So these macrophages are going to release chemokines, which is going to call a couple cells to the wound. It's going to call neutrophils, it's going to call new macrophages, and it's going to call mast cells. So mast cells are going to be around, and what mast cells do is secrete histamine. This is a really important one. So mast cells are secreting histamine, and histamine is going to dilate your blood vessels. And if you remember the word vasodilation, hopefully from last chapter, that's what histamine does. It triggers vasodilation. And when those blood vessels dilate, so when they go from this sort of to this, it's going to allow more blood to rush towards the wound, which is going to be able to carry more proteins. It's going to be able to carry more clotting factors. It's going to be able to carry more leukocytes to help fight off foreign substances. So that's going to be really important. So remember, mast cells secrete histamine to dilate blood vessels. And this is also why you see inflammation of a wound. If like you see red spots around a little cut that you have, that's because of the histamine. So the next step is going to be neutrophils are going to be recruited to kill pathogens. So a bunch of bacteria maybe are entering this wound. These neutrophils, which are the really fast responders, if you remember, are really good at coming in and eliminating those bacteria quickly. Then the last step is new macrophages are going to form and secrete more and more of these cytokines. Remember, chemokines is just a type of cytokine. So you can use those almost interchangeably, but chemokines is pretty specific up here. But so anyway, new macrophages form and will secrete more cytokines. And then as the wound heals, this response will lessen and lessen. And then once you're all healed up, then this will end. So everything before this point, we've been talking about innate immunity. We're going to switch gears now and we're going to talk about the adaptive immunity, which is antigen-specific response. So for the adaptive immunity, we need to be able to detect the antigens. So we're going to use antigen detection is going to be the first process. We're going to have antigen presentation, which means we're going to show this antigen to cells that can sort of do something about it. And then we have the lymphatic response, which is going to be our lymphatic cells sort of deciding what to do. And then we're going to have memory formation, where we're going to try to basically build up a defense in case that particular pathogen comes back. So the lymphatic response implies that we're talking about the lymphatic system. So the lymphatic system is important for your immune system as well as your circulatory system. So there's this fluid called lymph, L-Y-M-P-H. So lymph is a fluid that picks up toxins or microbes, and it's going to pick these up from your tissues, and it can carry them to lymphatic organs. And these lymphatic organs, which would be like, so it's going to take these toxins to lymph nodes, or it could go to the spleen, which is another big lymphatic organ. And it's going to travel here through lymphatic vessels. So it's sort of responsible for once antigen detection happens and is presented, we're gonna sort of funnel these through our lymphatic system to get them somewhere where we can deal with them. So to start, we have antigen detection and recognition. So we have two big types of cells, and these are subtypes of lymphatic cells. We have B cells and T cells. So these are both lymphatic cells, right? So we have B cells, which can bind antigens directly. And remember, antigens were little proteins on the pathogen. And then T cells, though, require an antigen-presenting cells, or APCs. And you may remember we talked about one of those on the last page with the innate system, which was our dendritic cells. So your dendritic cells, we also said that's the bridge between innate and adaptive immunity, and this is why. Because it's going to be able to present antigens to T cells. And it does that, anything that is an antigen-presenting cell needs to have something called a major hold on, let me Google this real quick, because I always forget what MHC stands for. Major histocompatibility complex. So MHC is major histocompatibility complex. And this is something this is going to be present on any antigen presenting cell. And basically what it means is the antigen presenting cell, let's say right here, is going to have a little binding site for the antigen. So let's say that this is the antigen and this is the pathogen. So this B cell can bind here, or sorry, this antigen presenting cell can bind that antigen. And when it does that, it's going to reconfigure a little bit and sort of show just this one part of a peptide, which is a protein. And this is the MHC. This is the major histocompatibility complex. And this is what is going to be recognized by the T cells. So remember, B cells bind directly. T cells need a antigen presenting cell. So underneath T cells, we have two subtypes. We have cytotoxic T cells and helper T cells. So cytotoxic T cells, as the name kind of implies, can release toxins to kill infected cells. So cytotoxic T cells can kill infected cells. Another thing to associate with T cells is CD8+. And I would just put this down on your note sheet, to be honest. So CD8+ is cytotoxic T cells, which kill infected cells, and these cytotoxic T cells need to bind to MHC1s. You also have helper T cells, which are CD4, and helper T cells activate other immune cells. So helper T cells will actually activate cytotoxic T cells, or they could activate B cells. And there are two types of helper T cells. You have Th1 and Th2. Th1 is going to activate cytotoxic T cells, so it's just going to help out by activating these. And then Th2 activates B cells. So it's going to go and activate those. All helper T cells are CD4, and they all need to bind to MHC2, so major histocompatibility complex 2, which is a different form, a different peptide. And so once these B cells are activated, they secrete antibodies. And we'll get into that on this page. So B cells have these B cell receptors that directly bind antigens and then present that MHC2. So we have a B cell, and the B cell has the B cell receptor right here, the BCR. And that's going to be able to bind to this antigen. And when it does that, it will present the MHC2. And then the MHC2 is going to sort of be a signal flag for those Th2 helper T cells. So the Th2 helper T cells, so we have a little T cell, can then bind to this MHC2. And when it does that, it's going to release cytokines. And cytokines do two things. They recruit other immune cells, and they activate this B cell to release antibodies and to proliferate, or to start cloning. So we had the B cell receptor binded the antigen and then presented an MHC2 peptide, which alerted these Th2 helper T cells, which then bind to that MHC2 and release cytokines to activate the B cell. And then the activated B cell is going to secrete antibodies and undergo rapid division. So antibodies are these little Y-shaped proteins with, they have light chains and heavy chains. And antibodies can bind to an antigen to mark them for destruction or to neutralize them or to help kill them. So antibodies are really, really important, and they are secreted by B cells. So B and T cells, all of the lymphocytes, they have their receptors are unique to a very specific antigen. So up here we had this receptor, right? This was only for this specific antigen from this specific pathogen. So you might have a bunch of B cells that are really good at recognizing the flu. You might have some other ones that are good at the common cold, and so on and so on. So just know that all of these lymphocytes are specific to one antigen, but we have sort of a library. So we have a ton of these B and T cells so that we have diversity in these receptor types, which means we have diversity in the types of antigens we can recognize. Remember up here I said that B cells, when they get activated, they start rapidly dividing. So replication of this specific B cell that made this specific antibody for this specific antigen creates what are called memory B and T cells. Memory B and T cells mean that the second time this pathogen comes in, we are going to be able to recognize it really fast. Whereas with this process, we maybe didn't have as many B cells for this antigen. It took a long time to recognize it, but eventually we got there. But then once the body goes, okay, we need to watch out for this antigen because it's going to be a problem for us. So it's going to create memory B and T cells, which are just going to wait. They're just going to wait for this antigen to show up again. And that is why vaccines work. And that's why, hopefully, that's the main reason you don't get sick back to back very often with the same sickness, is because your memory B and T cells are able to recognize pathogens quicker, the specific pathogen that it has receptors for. That's memory cells. So next I want to talk about the destruction of pathogens. So there are two ways we destroy pathogens in the body. There's the humoral response and the cell-mediated response. The humoral response is going to be driven by Th2, so helper T cell 2, and B cells. So B cells, the humoral response is B cells are going to release antibodies that act in the blood and the lymph. Notice that this is not acting on individual cells, it is acting on antigens that are in the plasma or the lymph. So these antibodies can mark these antigens, and therefore the pathogen, for destruction. And then we also have our leukocytes, our white blood cells, our neutrophils, our eosinophils, and our macrophages to help kill these things. So that's the humoral response. It's very much extracellular. So it's not going to be targeting specific cells. It's going to be targeting the things that are just floating around in the plasma outside of the cells. And so next we have the cell-mediated response. So the cell-mediated response is the attack on cells that have been infected. So these antigens want to get into our cells, and sometimes they do. When that happens, we are going to look to our Th1 helper T cells and our cytotoxic T cells to take care of that. So Th1, so helper T cells, are going to activate the cytotoxic T cells using cytokines. And those cytotoxic T cells are going to attach to infected cells. So if we have an infected cell with a bunch of pathogens, an activated cytotoxic T cell can attach to this cell and can inject toxins into this cell. It can inject toxins into this cell that cause apoptosis. And remember, apoptosis from last term a little bit, we briefly talked about it, means the bursting of the cell. So we're going to burst this cell, and the antigens are going to go out into the plasma where neutrophils can come and eat them up and eliminate them. So that is the cell-mediated response. All right, just a little bit more here. We have antibodies and their functions. So antibodies can work in 4 different ways. We have opsonization, which means it's going to tag the antigen for phagocytosis. We have neutralization, which means it's going to essentially just stop the antigens from being able to infect the cells. Maybe the Y-shaped antibody binds to an important binding site for the antigen so that it can't bind to our cells. We have agglutination. Agglutination means we're gluing pathogens together. We're creating these clumps of pathogens that make it harder for them to do their job and really easy for a big old macrophage to come in and eat it up. And then antibodies can also work with the complement. So one other thing antibodies can do is poke holes in the membranes of pathogens. And when we poke those holes in the membranes of pathogens, we can insert these things called attack proteins into the pathogen membranes. And what attack proteins do is they're essentially just like glorified aquaporins, and they force water into the pathogen, and it gets bigger and bigger until it bursts. So those are the 4 key functions of antibodies. And the last thing we want to talk about just real quick is going to be vaccines. So vaccines provide a first exposure to dead antigens or toxins. So what that does is it gives your B cells, your adaptive immune system, something to recognize a certain pathogen. So when you get your flu vaccine, you are exposing your immune system to flu antigens, but not live pathogens. So your T cells and B cells can still go through the recognition process, and this is ultimately going to create memory lymphocytes, which were those memory B cells and memory D cells. And then when we have a population, the more people that get vaccinated grants what's called herd immunity, which is essentially protection from the pathogen just because such a high proportion of people are immune to it. So what that would look like on a graph is you might have your vaccine elicits this first response from the immune system, and then the next time that this pathogen is presented, maybe when you actually get the flu, you can get a much stronger second response. So this is the second response, this is the first response. And so the second response is much stronger. We have much more, we have these memory B cells and T cells, we have antibodies ready to go. We have the recognition is already done for us, basically. All right, so that's just sort of a summary of the immune system, and I will see you guys in the next video.